Welcome to ONCOmmunity, the molecular oncology podcast by OncoDNA. After talking about large panel testing in molecular oncology in a previous podcast, today we are going to take a look behind the scenes of a molecular pathology laboratory. Professor Michael Hummel, head of molecular pathology at the Charité Hospital in Berlin, is here with us today. Welcome.
Ja, auch ein ganz herzliches Willkommen von meiner Seite in die...
Welcome everyone. I'm happy to have the opportunity to share the experience we have acquired in using NGS in pathology over the last 6 years, and I'm looking forward to discussing this with you today.
Und ich bin gespannt auf die Diskussion mit Ihnen.
In your pathology laboratory in Berlin, you perform comprehensive genome testing on cancer patients in everyday clinical practice. Could you please tell us which panels you use and how such tests can be carried out?
Ja, wie schon eingangs erwähnt, haben wir schon einige...
Yes, as mentioned, we already have several years of experience with gene panels. Of course, we started at a relatively small scale in 2014 with very small panels, and also the number of cases was significantly different compared to today. We now perform a wide range of different gene panels for various diagnostic questions, for myeloid diseases, for lymphoma diseases, for various tumours, and especially for lung cancer. We are one of the centres within the National Network for Lung Diseases, NNGM, and we offer a variety of solutions up to our customised large gene panels with 3 megabases and more than 600 genes. This helps us to find the right answers for various diagnostic questions, to offer various test applications for the different diagnostic requests, and we are therefore positioned quite broadly.
Die richtigen Antworten anbieten können für die Anfragenden und daher relativ breit aufgestellt sind.
Do you get enough DNA from tissue biopsies of solid tumours?
Ja, das können durchaus...
Well, that definitely can be a challenge, because obviously in the interest of the patient, the tissue samples should be as small as possible. We're talking about fine needle biopsies which contain a few cells only from which the DNA can be extracted. But we are quite successful. The laboratory staff has acquired a lot of experience over many years, so that in the vast majority of cases we can obtain sufficient DNA. Nor should we forget the world of RNA in order to ultimately make a reliable statement about the mutations or the transcriptome of the relevant patient.
Aussage zur Mutation oder zum Transkriptom der Patienten geben zu können.
So how fast and how feasible are the sequencing workflows?
Gute Frage. Also die Workflows sind...
Good question. The workflows that we use are based on Thermo Fisher technology and are partly automated, meaning we set up robotics-based processes for the library preparation and some other steps, which clearly facilitates the work of the technical staff and also allows us to handle the high sample throughput in the first place. Therefore, the actual workflow with the Thermo Fisher platforms is quite well described. We can expect significantly higher level of automation in molecular diagnostics in the future. How fast are we? So, the system runs for about 1 day. The sample preparations take another half a day. We need about a day and a half from DNA to getting the results. The raw data, and then of course we have to interpret the data that we have collected by drawing up a report in the end. Here, the most important results are summarized for the inquiring physicians to then support their clinical decisions. Those are our processes. I would say that the majority of cases are processed within 5 days.
Entscheidungen zu unterstützen. Das sind unsere Abläufe. Ich würde sagen, das Gros der Fälle ist innerhalb von 5 Tagen bei uns durch.
What is the actual outlay for you in terms of time and materials as a laboratory? Is the whole thing cost efficient?
Sie stellen gute Fragen. Die Frage wirtschaftliche Darstellbarkeit. Wir haben in Deutschland...
Those are good questions. The question of cost efficiency. You have to consider that there are various accounting systems in Germany. For inpatients in a hospital like the Charité in Berlin, there is actually no additional reimbursement for molecular diagnostics from the health insurers, which means that everything is billed via the treatment costs. These DRG codes are relatively old and usually do not include molecular diagnostics. The hospital has to do a mixed cost calculation to prevent shortfalls. For external submissions from the outpatient sector, we have the so-called EBM catalogue, which enables us to bill according to the corresponding services. The situation is, of course, more favourable here, as we are able to cover the costs of the patient's cases that we analyse to a reasonable extent. And then we also have privately insured patients who order a certain analysis. A cost-effective and cost-covering analysis is always possible in such cases. Last but not least, something that is becoming increasingly important is patients who are examined under a so-called special contract or selective contract with health insurers. Bills are settled directly with the health insurer and can, and this is really important, also cover inpatients.
Die Abrechnung direkt mit der Krankenkasse und kann auch, und das ist wirklich sehr wichtig, auch die stationären Patienten umfassen.
You're talking about patients. For which cancer patients do you use these comprehensive molecular genomic tests as a standard diagnostic tool?
Wenn wir von Standard sprechen, dann müssen wir...
When talking about the standard diagnostics, certainly we must pay attention to the approval conditions. There are biomarkers, molecular biomarkers, that are associated with an approved licensed therapy. And in practical terms, that's the basic information we have to provide. These examinations are defined by the therapy approval, and the license covers a few tumour entities, such as lung, colon, skin, cancer, and some more. There is currently only one biomarker that can be used across virtually all tumour entities. These are NTRK fusions. The official approval states no correlation with a specific tumour entity, so the situation can be summarised as follows. So far, there are relatively few tumour entities treated based on NTRK, but the focus lies on the main tumour entities. We will soon be adding more tumour entities like prostate cancer and presumably breast cancer, where the analysis can also be carried out on a molecular basis.
Weitere Tumorentitäten hinzubekommen, wo die Analytik entsprechend auch molekular durchgeführt werden kann.
So the oncologists send their requests for laboratory tests and genomic tests to you. Do you give them any support? Do you provide training for oncologists?
Als Hilfestellung geben wir natürlich.
Yes, surely. We provide assistance, especially for those with less experience, who also send us their test requests. We are back to the subject of inquiry. If we look at the inquiry and think, maybe we could address this or that question better by running a different test, or maybe the request is a bit vague, then we are already involved in the first step, i.e., when the inquiry is defined and we provide practical advice. The whole topic becomes particularly important with regards to the results. How do we deal with that? As pathologists, we are, of course, not the treating physicians, but the diagnosing institution. And for large gene panels, i.e. those which go beyond approved biomarkers, we and many other institutions organise what are known as molecular tumour conferences or molecular tumour boards. Here, the results of our genomic tests are discussed interdisciplinarily and in the context of clinical information, thus selecting the best therapeutic option and recommendation for the patient.
Therapeutische Option herausgesucht und empfohlen wird.
How relevant are such comprehensive molecular genomic tumour analyses in today's clinical routine?
Auch hier wieder, wie schon bei den
Here too, as mentioned for the accounting regulations, approved biomarkers have to be considered. Of course, those results from molecular diagnostics are crucial in determining further therapeutic decisions. That's why there are these approved biomarkers and therapeutic procedures. This group is clear as the decisions are completely predefined. It is more difficult, or let's say less precise, when using broad genomic testing that goes beyond the approved biomarkers. In this case, the benefit for the patient often depends on the availability of appropriate drugs that are suitable for the particular molecular changes of the relevant patient. They are not always available. In addition, there are, of course, advanced clinical trials or clinical studies that are actively recruiting. And the latter in particular is the decisive major part for me, where our patients with large panel testing have a chance to be enrolled into such clinical trials, where new forms of therapy are being established and developed for the benefit of the patient. And the identification of such patients for clinical trials is, I believe, a very, very important task, especially for molecular pathology departments at university hospitals, in order to identify the patients who qualify for inclusion in a clinical trial.
Die sich für den Einfluss in eine klinische Studie qualifizieren.
Clinical trials is one area, but how do you see the future and further developments in the field of molecular genomic tumor analysis?
Ja, ich bin natürlich kein Hellseher.
Well, I have no crystal ball, but looking at the developments over the last 6 years, that's how long we've been practicing molecular diagnostics with next generation sequencing. And what has been achieved during this time, it is not rocket science to predict that developments will basically continue. Sooner or later, we will also have to be able to offer very broad molecular procedures for a larger number of patients, and we will be working more and more in a pan-cancer fashion. In addition, we will have to deal with further approvals, which will of course expand the portfolio as such. And the obvious question here is what will happen with the liquid biopsies in the future? It started some time ago with one specific biomarker, the T790M EGF receptor mutation in lung cancer. The molecular pathology departments perform this test, but liquid biopsies are not requested very often. We will soon be able to detect PIK3CA mutations in breast cancer using liquid biopsy. It is developing much slower than originally expected, and we will see what the future holds. In the end, liquid biopsies may not be key to find all crucial therapeutic biomarkers, but may rather become an important application for monitoring the recurrence of cancer diseases.
Möglicherweise sind auch das Monitoring von Wiederauftreten von Erkrankungen wichtige Anwendungen für die Liquid Biopsies.
Thank you very much, Professor Hummel, for your insights and explanations. In the next episode of ONCOmmunity, the molecular oncology podcast by OncoDNA, we will discuss this topic from the perspective of molecular oncologists.